I'm Roland, like I said. I'm originally a biologist, trained as a biologist, and then I did media technology, which is kind of bridging between art and science and computer science. And I found it super interesting to find the kind of place where those two meet, where people use new technologies and combine them with biology and did creative explorations. And then I found the Open Wet Lab here years ago, which I'm now leading the BioHack Academy in. And it's great to find this place in Amsterdam, where you can do kind of biotechnology experiments, but then low-key or accessible.
That's amazing. I mean, we are literally in the heart of Amsterdam and the Waag in the market at the moment. And I would never have imagined that this place, this beautiful old tower, actually holds labs that does all of this cool stuff. So what is your motivation?
Well, motivation is multifold, I guess. One of the reasons is that it's great that people can do biology experiments outside of the lab, or at home, or in a community lab like this is, in essence. And also that the real citizens can be scientists too, you know, not like citizen science in the sense of data gathering for science, but really inquiry and doing your own research, which can be done both for scientific purpose or like for answering questions, or some people who use it more like in an applied sense, or in a design or art sense. But for Waag as well, it's also really about the critical thinking and about kind of being critical about new technological developments, because it's really believed that if you have more hands-on experience with these experiments or with these technologies, you can form an opinion at a different level than when you just passively discuss it. So that's also one of the reasons the Open Wet Lab is here.
Oh, that's wonderful.
Yeah.
So have you had any, I know that you do courses that do some, different things. Could you tell us more about that, actually?
Yeah. So at the moment I'm leading the BioHack Academy, which we're midway through at the moment. We're recording this, and this is this 10-week course where there's participants in Amsterdam and in partner labs worldwide that follow the course simultaneously, and it's basically about building your own lab and then using it for bio experiments, and then sharing your experiments and your designs, and kind of hacking the tools together. So every week we go through a new scientific tool, like we have do-it-yourself microscopes and incubators and PCR machines, and then people can choose to make them. But most importantly, they get a lot of lab time here, which is great. So that we start week 1 with laboratory safety, and they get drilled into how to work safe in a lab. And the people who never used to work in a lab before, right? And I learned pipetting and microbiology, and now I feel we have a group of people who are really skilled to work in this lab. So that's really cool.
Wonderful. So these people really have never worked in a lab before?
No.
Wonderful.
Yeah.
So can you talk a little bit more about the projects that you do? For example, I heard about PCR machines. Are you actually building PCR machines from nothing? You fabricate those things?
Yeah, so I can show you something as well. So at the BioHack Academy, we built these different devices. So here's, for example, a PCR machine, where you have the metal parts which heats up and cools down, right? And if you tubes in, and you get this reaction that amplifies DNA. But the idea is that these kind of machines normally are super expensive, so why not look at how they function and make them much more accessible and cheap?
That's wonderful.
Because we have the fab lab next door, which has all the machines, like to laser cut stuff. So we basically use these machines to build machines for the lab, which makes it much cheaper. But apart from, well, of course it does cost a lot of time and tinkering, but it's also more about the idea of knowing how a tool works and understanding the things you work with. It's also a challenge. At the Waag in general, there's one group of people who started hacking, but it was during the open nights we had. They started hacking with digital microfluidics. So it's a technology to move little micro droplets around using electrostatic forces, so with high voltage. And they started just with like photo print paper and silver ink and making these patterns, but now they're actually a little startup company that's growing and growing actually. It's from a good friend of me, Federico, they call DigiBio. So that's really expanded from hacking to doing. And we saw some more projects here, especially from designers. For example, upstairs they work a lot with bacteria dying, and then that's also started like as something small of a workshop, and then it got picked up and expanded.
So what is bacteria dying?
It's taking the pigments from bacteria and dyeing clothes with them.
Oh, wonderful! Wow.
Yeah,
so there are practical applications of what actually is being done here. There could be, basically.
Yeah.
What do you hope to achieve in the end, actually?
It's a good question. Also because my goalposts are changing all the time.
Yes.
No, I'm really inspired by seeing all the people developing very cool projects and like picking this up and taking this free place to develop these things, kind of from their own curiosity, following that and and developing things that either well, what I really like here, especially in the Open Wet Lab, is that people can work really with biology itself. I mean, nowadays there's a lot of kind of projects which are more speculative in a way about the future of biology, like paint a picture, but here people work with the wet stuff itself. And that's great. And how I hope it will develop. Well, one thing I'm really curious about is, like, you have a background in life science yourself, right?
I do.
Yeah, and many people in life sciences then in the end go do something else, but they still could also use a lab. Maybe want to have some side project in a lab, and I think this could be some interesting thing in the future. Not only for people who did life science, but in general, because when you want to do a project and it's with a digital or it's with a computer, anybody has a laptop nowadays, but when you want to do something with life sciences, it's really hard to do, and community labs like this one can play a role in these kind of things.
Are there more labs in the country, in the Netherlands, that are like these community labs, for example? Are there more examples of this place?
Yeah, I mean, worldwide there's a couple of do-it-yourself bio labs. In the Netherlands, you have Blue City Lab in Rotterdam, in the old Tropicana, in the old swimming pool, where also a lot of sustainable companies are housed that kind of use each other's waste as a base for their product. That's the idea of a blue economy, but they also have a little wet lab. And you have BioArt laboratories in Eindhoven.
So I noticed from your website that there are actually courses. So it is a 10-week course, basically.
10 weeks.
What if somebody comes in here and has a completely new idea? Do you have the capability of facilitating this, perhaps?
Oh, we try. Now somebody came and she wants to decellularize plants and then use it to regrow organs as an idea.
Wow.
So then we start the conversation and start hacking, and people work in this space. Maybe up until some point you cannot use this lab anymore, but then maybe there's other labs in the Netherlands which you could use. So we also work, especially my colleague Lucas Evers, he works a lot with bioartists and also helps them work in different labs around the country. So for example, there was recently one that played a synthesizer with his neuron cells. And that was something that could not be done here, I think, because of the biosafety level for neuron cells. But then they used a lab somewhere else.
Fantastic. Yeah. What do you think is the application of what you're doing here? Can you perhaps use this technology and the protocols that you're using to build these machines? Can they be used in, for example, places where they cannot afford a PCR machine or a microscope or a hood, for example?
Yeah. So yeah, hacking sounds a bit
negative,
dark. I think I think most people have maybe a dark association, like a Mr. Robot type of computer hacking. But I think hacking can also be a positive thing, like meaning taking things apart and understanding how they work. And it also started as a positive word the first time it was used, as I understand it.
Oh really?
Yeah. It started with MIT, with people making puns at each other and like doing hacks. And then they wanted to do stuff that nobody understood how they did it. These were the hacks they did. I don't remember which one. Yeah. Like putting a car on top of a roof or something like that, weird stuff. No, but yeah, I mean the whole open source hardware movement in general is really picking up right now, also in places where they have less resources to use these technologies. So that's great.
Yeah.
Also of interest of different labs. For example, in Africa, I want to work with biology, and then these designs can come at hand, or maybe different designs, maybe just the principles, because it really depends on what you can get as well in these situations. I mean, here we are a bit in a luxury situation. You can order anything and it will arrive. Yeah. For the BioHack Academy, the age stretch is pretty big. I mean, I think from, I actually don't know how young the youngest is, but I'm guessing like from 18 to 70 or so. Oh, wow. So really diverse. And it's also fun because we work with different partner labs in different countries, and they have also very different participants. So so we have people now in Tokyo, in Seoul, and in New Mexico. Over in Albuquerque in the US, and they all have very different groups, which all follow the same course. So you can learn quite a lot from different perspectives. Yeah. And in general, some people do stay involved with the whole bio community, or continue their projects, or or keep coming back here. So yeah,
it's very inspirational for these people.
Yeah.
Nice.
Yeah, and it also is the case that we cram so much in this BioHack Academy that you're never going to get it finished anyway, because because there's all this device you can make with. It's 2 days a week, it's 10 weeks, but there's also workshops and practicals. So it's not the goal to make everything within those 10 weeks. Also because I want people to focus on their own curiosity in their own project. Mm-hmm. So if they do want to make everything, that means it's like a year-long endeavor, you know. But it gives a lot of inspiration to continue. And people do continue, like last year one participant was working on soft robotics, which is this new technology to make robots that are made of silicon. You fill them with air and then they float up, or like fill with air, yeah. And he's still working on that. So, and another one from last year was involved in a bio-art project here with somebody that did, with Spela Petric. It was Bart Peters. They did the lip reading of plants. They looked at the stomata, like on the side of a leaf, and they look like lips. And they put an algorithm on it, and they lip read the plants.
So what did the plants say?
Oh, and I also did interpreters. So like interpreters of. I don't know what they said, but I mean the whole idea of it's still an ongoing project, I guess. Wonderful. As well. Yeah.
Wonderful.
But,
tell us about one of your projects that you are particularly proud of.
Well, we did this one project here in the lab with Günther Seyfried. He's an Austrian bio-artist, with Günther Seyfried and two more people, I have to say, Federico Mufato and Hans-Jörg Petschko. But Günther has this whole series where he works with, he takes an image, he translates the pixels into DNA code, so every color, every pixel becomes certain combination of base pairs in DNA, and then he orders this DNA and he mutates it in some way in the lab. So you kind of get a Photoshop via the biolab. Genetic Photoshop. And we did this one project here called Return to Deelmoon, which we used then. So we took an image, we converted it to DNA, and we cut out a part using CRISPR, which is this new gene editing technology. But then we did it in vitro, which means without cells. So a cell-free experiment, which is why we also could do it here, because it's not an organism then. When you use genetically modification, but you don't use any organism. And we had this picture of this bull's head, originally from the Deelmoon area, which is near the current Bahrain. But this was the original picture, so once again, like all pixels were converted into DNA, and then we had it in a tube, right? We wanted to cut out this part, so then the hollow eyes of the bull would become would open up, kind of. It would be white eyes. And that was the goal, and the result in the end was this, so, which means
Fantastic. So
which
that
means
means,
it works.
It wasn't, there was conversion, obviously, in the right place, but there's also some other things that are happening.
Yeah, we pasted it in there, some other indel mutations and sequencing errors, I think, is well happening, because when the base pairs change, the color changes as well, because of the conversion algorithm. And then in the second experiment, we reflected upon how much off-target effects CRISPR has, so how much times it cuts wrong, and whether it's as precise as it is thought to be. So we used shorter guidance RNA, so of 12 and 15 nucleotides to create random cuts, basically. So here you see those results.
Very interesting.
And that was great. That was a challenge to do here, because of course we're here in the it's the open wet lab, right? So for example, we don't have gas, but we use a lot of the gas tanks that are there, and but we had to work super sterilely, because this is cell-free, and you work with RNAs, but the RNase enzyme is everywhere. So as well, the first time that it worked, afterwards it didn't work anymore, because I think our RNase degraded because of this RNase enzyme. So then we had to start again, making new guidance RNA, and we had to work really controlled. So we were very happy that it worked. And it also shows that you can really use a lab like this for molecular biology experiments, which is great. Yeah.
I mean, it's my mind is being blown right now because of all the applications that you guys can do with this kind of work. I mean, you're fabricating things. You are actually creating, changes in DNA using CRISPR in the lab right here.
Yeah.
I mean, the applications are endless, I would say.
Yeah. Well, except that we're not allowed to work really with cells.
Well, sure, but I mean, still,
at the
without
moment,
working with cells and just still being a low biological level lab, you're still managing to do all of these fantastic things. I think that is something which is, absolutely amazing to me.
Yeah, I think it's great too. And this is, for example, the gel box, also open source from IO Rodeo, which is like blue light and a orange filter. And here we saw it cut. So this was the fragment that cut into
Very nice.
2 parts. Yeah. So that was one of the most challenging projects I did here in this lab. But apart from that, we do a lot of workshops as well. So we did one workshop on CRISPR with the do-it-yourself CRISPR kit from the Odin. You know that one?
No,
it's
I don't actually. No.
It's a kind of biohacker company in California who sells CRISPR kits for a couple of $100, and then people can introduce this, I guess, ampicillin, or not ampicillin, but different antibiotics resistance in a E. coli bacteria, and then you can check on your plate whether it worked or not. But we're not allowed to do that until the fully end here, because we don't have we have a license for genetic modification, but not for this. We have to get a new kind of agreement for all experiments we want to do in this lab. We're still proud of being like one of the labs that has this GMO license, but it's still a long process to get that. So we did this experiment with CRISPR, but we went until the step that we could not legally continue, which is the heat shock. So we brought the plasmids and which contain the DNA, and then the cells into one tube, and then you had to heat shock it, and when it's heat shock, the cells take up the DNA. So we had this big discussion around the heat pen, like, are we going to do it, are we not going to do it? Kind of the ethical implications of CRISPR in general, like building upon this like one moment until all the other societal issues around it. And that was great, yeah.
Wow, fantastic.
Yeah. So we do quite a lot of workshops as well, like bringing up these issues. Yeah.
Okay.
Yeah, but this is very, this is very deep molecular biology, but we also have bio-artists here working completely different fields. Like here you see a photo of, if I say it correct, Jennifer, I have to look up the names, Cara Riley and Jennifer Willett. Yeah, Jennifer Willett and Cara Riley. And it's part of a bigger kind of series called Trust Me, I'm an Artist, where they had bio-artists do a performance in front of an ethical committee, because normally only science performs for an ethical committee or performs, sends in project and they get approval or not. But in this case, the performance was done every time by an artist like in different cities for different ethical committees. And here you have the one on the right. She works with fish and glitters, and about how we pollute the environment. And on the left you see the woman has a lot of incubators on her body, so she incubates some kind of swabs on her on her suit. So this is much more performative, and it's also being done here at the Waag. So to show you like the big breadth of projects.
Yeah. So it's not just performative; it's also informative, I would say.
Yeah.
That's
Yeah, and it questions the what we're doing.
Yeah.
I mean,
Wonderful.
as humanity. Yeah.